So, welcome class. So, we will start today with the continuing the same part that we were doing last time. That is your molecular biology methods, in which we have discussed quite number of methods yesterday, and today we'll be dealing with next part, that is sequencing. Okay. So DNA sequencing, the Sanger sequencing method. Here what you do is, in this method, this is quite old method identified by the Sanger himself. So what they do is, if this is the known reaction and you identify that till how long is your sequence is, what is your sequence is made up of, then you add DNA polymerase with nucleotides plus GGGTPs in low concentration. So these DDGTPs will attach to the last part of the C, and until then it will polymerize the whole reaction. So by this you will have number of different bands here, different required actions to be presented. So you have this template, and this is a primer, and this primer will go until where it could go, and then we will run them over the gel. Then after running the gel, we can see them with the different colors with the bioinformatics computational biology, then A, T, G, C, T, T, C, G, like this. And then we can confirm that this is the long sequence of our concerned party. Let me show you how we do it. Now we will discuss about next generation sequencing, which includes your different parameters, various technologies like Roche, Solid, Pacific Biosciences, Iron Torrent, Genome and Illumina. So like this. So each has its specific working of doing it. So like Roche, Illumina, and Solid, their chemistry is pyrosequencing, polymerase based, and ligation based. Whereas the amplification is done by the emulsion PCR, bridge amplification, emulsion PCR, and the most best one so far out of them is, even from the time point of view, Roche is the best. Even it's a bit cheaper, and cost per MB is a bit expensive as compared to the Illumina. So these are the various principles. Just go through them. I will show the respective videos for them. How do they work?
Next-generation sequencing, or NGS, is a powerful platform that has enabled the sequencing of thousands to millions of DNA molecules simultaneously. This powerful tool is revolutionizing fields such as personalized medicine, genetic diseases, and clinical diagnostics by offering a high-throughput option with the capability to sequence multiple individuals at the same time. Sanger sequencing, first developed in the 1900s, is the gold standard for DNA sequencing, and it is still used today extensively for routine sequencing applications and to validate NGS data. It utilizes a high-fidelity DNA-dependent polymerase to generate a complementary copy to a single-stranded DNA template. In each reaction, a single primer, complementary to the template, initiates a DNA synthesis reaction from its 3' end. Deoxynucleotides, or simply nucleotides, are added one after the other in a template-dependent manner. Each reaction also contains a mixture of 4 dideoxynucleotides, one for each DNA base. These dideoxynucleotides resemble the DNA monomers enough to allow incorporation into the growing strand. However, they differ from natural deoxynucleotides in 2 ways. 1. They lack a 3' hydroxyl group, which is required for further DNA extension, resulting in chain termination once incorporated in the DNA molecule. And 2, each dideoxynucleotide has a unique fluorescent dye attached to it, allowing for automatic detection of the DNA sequence. As a result, many copies of different length DNA fragments are generated in each reaction, terminated at all of the nucleotide positions of the template molecule by one of the dideoxynucleotides. The reaction mixtures are loaded on the sequencing machine, either manually onto slab gels or automatically with capillaries, and are electrophoresed to separate the DNA molecules by size. The DNA sequence is read through the fluorescent emission of the dideoxynucleotide as it flows through the gel. Modern-day Sanger sequencing instruments use capillary-based automated electrophoresis, which typically analyzes 8 to 96 sequencing reactions simultaneously. Next-generation sequencing systems have been introduced in the past decade that allow for massively parallel sequencing reactions. These systems are capable of analyzing millions or even billions of sequencing reactions at the same time. Although different machines have been developed with various differing technical details, they all share some common features. 1. Sample preparation. All next-generation sequencing platforms require a library obtained either by amplification or ligation with custom adapter sequences. 2. Sequencing machines. Each library fragment is amplified on a solid surface with covalently attached DNA linkers that hybridize the library adapters. This amplification creates clusters of DNA, each originating from a single library fragment. Each cluster will act as an individual sequencing reaction. And 3. Data output. Each machine provides the raw data at the end of the sequencing run. This raw data is a collection of DNA sequences that were generated at each cluster. The differences between the different next-generation sequencing platforms lie mainly in the technical details of the sequencing reaction and can be categorized in 4 groups: pyrosequencing, sequencing by synthesis, sequencing by ligation, and ion semiconductor sequencing. In pyrosequencing, the sequencing reaction is monitored through the release of a pyrophosphate during each nucleotide incorporation. The released pyrophosphate is used in a series of chemical reactions resulting in the generation of light. Light emission is detected by a camera, which records the appropriate sequence of the cluster. The sequencing proceeds by incubating one base at a time, measuring the light emission, if any, degrading the unincorporated bases, and then the addition of another base. This technology is capable of generating large read lengths, much comparable to the read length of Sanger sequencing. However, high reagent cost and high error rate over strings of 6 or more homopolymers have reduced its applications. For more details on the technical aspect of this technology, please visit our knowledge base at the link provided in the description below. Sequencing by synthesis utilizes the step-by-step incorporation of reversibly fluorescent and terminated nucleotides for DNA sequencing and is used by the Illumina NGS platforms. All 4 nucleotides are added to the sequencing chip at the same time, and after nucleotide incorporation, the remaining DNA bases are washed away. The fluorescent signal is read at each cluster and recorded. Both the fluorescent molecule and the terminator group are then cleaved and washed away. This process is repeated until the sequencing reaction is complete. This system is able to overcome the disadvantages of the pyrosequencing system by only incorporating a single nucleotide at a time. However, as the sequencing reaction proceeds, the error rate of the machine also increases. This is due to incomplete removal of the fluorescent signal, which leads to higher background noise levels. Our NGS, an introduction knowledge base, provides more technical details about this technology. Sequencing by ligation is different from the other 2 methods since it does not utilize a DNA polymerase to incorporate nucleotides. Instead, it relies on 16 octamer oligonucleotide probes, each with one of 4 fluorescent dyes attached to its 5' end that are ligated to one another. Each octamer consists of 2 probe-specific bases and 6 degenerate bases. The sequencing reaction commences by binding of the primer to the adapter sequence and then hybridization of the appropriate probe. This hybridization of the probe is guided by the 2 probe-specific bases and upon annealing is ligated to the primer sequence through a DNA ligase. Unbound oligonucleotides are washed away, then the signal is detected and recorded. After that, the fluorescent signal, along with the last 3 bases of the octamer probe, are cleaved, and then the next cycle commences. After approximately 7 cycles of ligation, the DNA strand is denatured and another sequencing primer, offset by 1 base from the previous primer, is used to repeat these steps. In total, 5 sequencing primers are used. The major disadvantage of this technology is the very short sequencing reads generated. Ion semiconductor sequencing utilizes the release of hydrogen ions during the sequencing reaction to detect the sequence of a cluster. Each cluster is located directly above a semiconductor transistor, which is capable of detecting changes in the pH of the solution. During nucleotide incorporation, a single hydrogen ion is released into the solution and it is detected by the semiconductor. The sequencing reaction itself proceeds similarly to pyrosequencing, but at a fraction of the cost. Please view our knowledge base for further details on ion semiconductor sequencing and the sequencing by ligation techniques. In order to be able to showcase and compare the different technical aspects of each of the above technologies, the number of coverage that each run generates when sequencing the whole human, mouse, Arabidopsis thaliana, and E. coli genomes are calculated and presented here. The presented data is based on the most powerful machines of each technology. Further details can be found on our knowledge base. For whole genome sequencing data to be useful, a minimum of 30 times coverage is required. As it can be seen, the pyrosequencing method is only able to sequence the E. coli genome at enough coverage to result in valid data. The sequencing by synthesis method, which is the most popular method currently on the market, is able to generate hundreds of coverage per run. In fact, with this machine, it is possible to sequence 15 individuals within 3.5 days. The sequencing by ligation method also generates enough coverage for all genomes to be used. However, it isn't capable of generating nearly as much output as the Illumina HiSeq machines. The ion proton machine is used mostly in clinical settings because it is able to provide a sufficient size output within 2 hours. ABM offers a wide range of next-generation sequencing services. These include whole genome sequencing, exome sequencing, RNA sequencing, disease panels, lane rentals, and much more. To be able to access our services, please visit our website at www.abmgood.com, and from there, click on the NGS Sequencing Services link. This will load our NGS service webpage, which details all of our available services. Clicking on a service of interest will showcase the technical details, pricing, and bioinformatics solutions that are related to that particular service. Please leave your questions and comments below, and we will answer them as soon as possible. For more information, please visit our knowledge base at the link provided below. Thank you for watching.
I hope that was clear from the NGS point of view. So these are the applications. They could be helpful for genomes, resequencing, human exons, long RNA profiling, chip sequencing, DNA methylation, polysomal RNA, and they cost around 1000 genome per sequence. Now let's discuss about method of study gene function. So to study the gene function, some basic principles and nomenclature here. So on the green side, what you're seeing is that from mutant, your clone gene is produced with the DNA sequencing. Then the protein localized is being produced. Then on the red side, database search to identify protein coding sequence with gene inactivation is being produced there. So there are four types of research, that is in vivo, ex vivo, in vitro, and in silico. The in vivo will be in living organisms like bacteria, yeast, monkey, Christmas tree, whereas ex vivo is in organic explant, example tissue biopsy, whereas the in vitro is for the reagent tube synthetic setup, whereas the in silico is computer based simulations or data analysis. So advantages of working with cultured cells over intact organisms. So more homogeneous than cells in the tissues, can control experiment conditions, ethically non-controversial, and we can drive cell culture from bacteria, yeast, vertebrates, plants. In the vertebrates, we can use models from the mouse, human, rat, hamster, whereas in plants, we can use Arabidopsis thaliana. So growth of microorganisms in culture. So these are some examples shown here in the below images. The bacterial colonies and yeast colonies. The bacterial colonies is having Escherichia coli and the yeast having Saccharomyces cerevisiae. So having rapid growth rate, simple nutritional requirement, can be grown on the semi-solid agar, and mutant strain can also be for the replical plating. So growth of animal cell culture in cells are shown in the bottom, which require essential amino acids, vitamins, salts, glucose, and serum, and mostly grow on the solid surfaces, like blood cells grown in the suspension. So single mouse cell look like this. On the left side, in the middle, is a colony of human cells. On the right is many colonies in the petri dish. So primary cell line and primary cells and cell lines. So at the bottom you can see there are two types of cell lines, human cell line and mouse cell line, and their growth rate of culture on the left side, and on the y-axis is the cell generations. So growth rate of culture is shown in the phase 1, phase 2, and phase 3. With the cell strain, first it's being like linear in the human cells, then it's stagnant, then further it started to give cell senescence. Whereas in the mouse cells, the culture initially has a loss of growth potential, but after some time for senescence, 30 days, it goes for emergence immortal variant cell line. So that's how that major difference between a human cell line and mouse cell line. Here we can see some fibroblasts which are cultured, which are primary or immortal and differentiated further. We call them as NIH or 3T3 mouse embryo fibroblast. And further we can see some undifferentiated cells, differentiated cells. Here on the left side is the undifferentiated cells of fibroblasts. But when they get differentiated, this is with the dye, a red dye here, we can see proper function as a fibroblast. They are coming together and good to go for. So some of the generation. So today our task is to finish with molecular biology class here. We will just finish with that part mainly. So we will discuss things very slowly here from now onwards. That is the generation of transgenic mouse and knockout mouse. This is very hot topic. There's a Charles Rivers, many companies who are working on the making of your knockout, knock-in, transgenic, conditional, tissue-specific knockout. Many mouses in the field of molecular biology. So, like the loss of function is known as knockout mouse, or we can also remove the function by siRNA transfection. Whereas we can make the gain of function, we call that mouse as a transgenic mouse. Whereas the flavors could be conditional or tissue specific based on that. Then there are mutation type and causes. So, so as we know, there's called thing called genotype and phenotype. Genotype includes the genetic makeup, where the phenotype includes the function and physical appearance. So normally in the wild type, organisms may develop changes termed mutations in the DNA sequence, where the organisms that develop such mutations are termed as mutants. So there is a haploid and diploid. Haploid organisms has a single copy of each chromosome, and its phenotype is consequence of that one copy. And this diploid has two copies of each chromosome, thus two copies of each gene. So we can see that alleles may be dominant or recessive. So diploid genotype here and the diploid phenotype. So diploid genotype includes these wild type here, and the mutants having the dominant genes are yellow, and the wild type as being recessive 1 and 2 is being mutant here. And some genotypic linked diseases, so like sickle cell anemia, which is happening because of abnormal hemoglobin, which cause deformation of your red blood cells and clogged your in the capillaries, and resistance to malaria. So this is mostly found in the sub-Saharan African region. There it has been our region. Then there is cystic fibrosis that is happening because of CFTR, defective chloride channel in epithelial cells, lead to excessive mucus in lungs. Then phenylketonuria, which is a defective enzyme in phenylalanine metabolism, um, results in excess of phenylalanine amino acids, leading to mental retardation unless restricted by diet. Then Tay-Sachs disease, defective hexamide enzyme leads to accumulation of excess sphingolipids, lysosomes of neurons, impairing neural development. So these were all autosomal recessive genes, and there's autosomal dominant also. So now autosomal dominant includes your Huntington disease and hypercholesteremia. So in the Huntington disease, it includes defective neural protein misassembled into aggregates cause damage to neural tissues, whereas the hypercholesteremia, defective LDL receptor leads to excessive cholesterol in blood and early heart attacks. Now comes the X-linked recessive, the X-linked diseases, DMD and hemophilia A. In the Duchenne muscular dystrophy, your defective cytoskeletal protein dystrophin leads to impaired muscle functions, and in hemophilia, defective blood clotting factor 8 leads to uncontrolled bleeding. So each has its own function, its own way of doing the disease, from the autosomal recessive, autosomal dominant, and X recessive. Then segregation of genotype linked diseases in the next part. So here is the autosomal dominant Huntington disease, in which your male is affected, female is not. So male and females will have affective and non-affective. Whereas the autosomal recessive cystic fibrosis, which will have CFTR as a carrier, and the males and females are being affected here on the left side, and the rest are carrier and non-carrier. And on the other side, X-linked recessive Duchenne muscular dystrophy. Here's your female is the carrier, so your males are being affected in this case. Another examples are mutants in Drosophila, that they're having this red eye, and the white eye having 2 wings and 6 wings. And also in the gene replacement and transgenic mouse, either we can inactive the gene, we call that as gene knockout, or we can add one gene to increase the effect of that gene, or we can do also mutagenesis in order to create a mutant. So some of the ways to produce your knockout mutations in embryonic stem cells before you put them into the mice. So specific targeting of gene of interest. So on the left side, you can see formation of embryonic stem cells carrying a knockout mutation. Here the gene X is replaced with the construct, and then the homologous combination or non-homologous combinations are made with the ESL line. Then the ESL line, when is added, then there is a crossover is taking place with other genes. This is the gene targeted insertion. So mutation in the gene X cells are resistant to G418 and gynocovala. Whereas in this case, random insertion has been done, so no mutation in the gene X cells are resistant to G14, but sensitive to gynocovala. Further gene knockout in the mouse, how we can do it? We inject this embryonic stem cells, which we have here factored with our trans gene X. Then we put them into the blastocyst of 4 to 5 days. So here we have the recipient is a black mouse and the donor is a brown mouse. So surgical transfer leads to a pseudo-pregnant female and foster mother, possibly progeny will be mixture of both, chimeric and black. And the selected chimeric will be crossed with the black mouse again, leading to produce your progeny with the embryonic stem cell from the germ cells and the black one. Then we screen for the brown progeny, heterozygous, then mate with the heterozygote, and the screen progeny DNA to identify homozygotes. At the end, what you're seeing is the knockout mouse. And this is a bit complex. LOXP mouse model, Cree and LOXP actually. When I was doing my master's, I remember Professor Stephen Offermans, very famous scientist from the Max Planck Institute in the Department of Pharmacology. He's a group. He's like had the director of the Max Planck. So he's the one who created your LOXP mouse, you know. So he doesn't create, but they were famous for creating these mouses. Um, so there's a LOXP mouse and there's a Cree mouse. The LOXP mouse have all endogenous genes with LOXP sites flanking exon. So here LOXP, and there's a heterozygous for gene X, a knockout. All cells carry CRE gene here. Then cells expressing CRE, Cree gene, and here on the left cell not expressing CRE gene. So gene function is normal, so LOX, but here the cell expressing the Cree gene, they are crossover like this, 1, 2, then 3, and your Cree protein is in between. Then gene function, we remove this 2 part and this one, and then gene function is disrupted in this case. So we have all cells carrying the LOXP modified gene X, one copy of gene X knocked on Cree genes in this case. And we can do also gene silencing with small interference RNA. That is, we have a gene of interest with the sense and the antisense transcript, right? Then we bring them together with the dicer, we create this siRNA. Then we inject them, and then we design to make the expression of the specific protein or RNA will not work. And production of transgenic mice to increase the production of your thing. So you inject foreign DNA into one of the pronuclei, then you put them into the foster mother. Then about 10 to 30% of offspring contains injected foreign DNA. Then mice expressing DNA is bred to continue down DNA in the germ line. So this is the example of rat having the growth hormone being grown up higher by flanking it in this regulatory sequence with the rat GH gene to be present there. So let's see some videos about how this LOXP and everything works out, how we do this in practically way. Let's check them out now.
Gene knockout is a common technique in studying gene function. In this practice, the gene of interest is disrupted by altering the cell's genomic DNA. Any phenotype that results from gene knockout gives insight into the function of the disabled gene. Conventional knockout experiments are frequently achieved in animals, but need a huge investment of time, money, and labor. Therefore, these experiments are usually restricted to well-funded labs or core facilities. This has presented a large hurdle for researchers exploring gene function. The breakthrough of CRISPR technology has turned the tide and made gene knockout a much more accessible tool for many more research labs. The key revolution brought about by CRISPR is the creation of double-stranded breaks sequenced specifically. With CRISPR, scientists can create double-stranded breaks at virtually any site in the genome. To repair the double-strand breaks generated by CRISPR, there are two main mechanisms: non-homologous end joining and homology-directed repair, also known as homologous recombination. With non-homologous end joining, the ends of the double-strand breaks are simply reconnected together, and this process is highly error-prone, introducing mutations, insertions, and deletions, and in many cases resulting in gene knockout. Homology-directed repair, on the other hand, utilizes a repair template, where desired changes are flanked by left and right homologous sequences. Through homologous recombination, the desired changes are incorporated into the genome. The CRISPR knockout knock-in kits utilizes the homology-directed repair system. To plan an HDR-backed knockout experiment using plasmids, the researcher needs to create the following constructs. 1. A plasmid coding for the gRNA, targeting the intended site for double-stranded cleavage. 2. A plasmid coding for the Cas9 protein. And 3. A plasmid serving as the repair template, with a designed sequence flanked by the homologous arm region of the cleavage site, or donor vector. Origin has created pre-designed kits to make knockout experiments even easier. For each gene locus, we have created a specific kit. The first two elements are provided in a single all-in-one vector. The gRNA vector, targeting the start of the coding sequence, was cloned into a vector containing the expression cassette for Cas9 protein. The donor vector was also created with a knock-in GFP puromycin cassette, flanked by the homologous arms of the chosen cleavage site. This permits the selection of knockout cells via puromycin marker. The promoterless GFP provides additional utility to monitor any upstream promoter activity that might be present. To start your knockout experiment, simply co-transfect a gRNA vector with a donor DNA plasmid into the cells. The Cas9 enzyme will cut a double-stranded break near the start codon, and through homologous recombination, the functional cassette will be integrated into the genome, replacing target gene. In the end, 3 events are achieved. 1. The target gene is knocked out through gene disruption. 2. GFP will be driven by the endogenous gene promoter, providing added information of promoter strength. And 3. The puromycin marker under the PGK promoter is integrated into the genome, enabling the selection of successfully edited cells. The protocol starts with co-transfection. There will be 3 separate experiments. One of the 2 gRNA vectors co-transfected with the donor vector, scramble control with donor vector. After transfection, the cells must be passaged for about 20 days before puromycin selection, as the donor vector alone before genomic integration also provides puromycin resistance. Culturing the transfected cells for 20 days ensures that the episomal form of donor vector is lost. This diagram shows the details of transfected cell passaging prior to puromycin application and selection. For HEK 392 cells, 48 hours post-transfection is considered G1 cells, at which point the cells are split 1 to 10 and grown for 3 more days. At G2, CRISPR-targeted cleavage and gene editing should be finished, but the percentage of edited cells will be small. You can perform optional genomic DNA analysis using G2 cells. If the data are negative, you can redo the analysis after puromycin selection. After G7, apply puromycin selection using the doses appropriate for your cells. To summarize the CRISPR knockout knock-in protocol described so far, Step 1. Co-transfect one of the gRNA vectors with the donor vector, along with the appropriate controls. Step 2. Passage transfected cells for around 20 days. Step 3. Apply puromycin selection and isolate individual cell colonies. Please note that the drug selection doses need to be determined by kill curve for each cell line. 5 days after puromycin selection of transfected HEK 293 cells, almost all cells in the 2 control transfections died. However, as expected, the co-transfection with the gRNA construct produced puromycin-resistant cells. The fourth step is to analyze the puromycin-positive cells to detect the knockout. If you have a good protein-specific antibody, you can use Western blotting to evaluate the knockout. It is best to use this method after isolating single colonies. Another method is genomic PCR to verify the correct GFP puromycin integration in the genome, followed by sequencing to confirm the integration. To amplify only the edited allele, design PCR primers following these rules. The forward primer should be upstream of the 5' end of the left homologous arm of the donor vector, and the reverse primer should be in the GFP region. In this example, knockout ATG5 gene was conducted by the protocol. Genomic PCR products of the correct size were amplified from the gRNA constructs, plus the donor co-transfected cells, and not from the scramble control transfected cells. The PCR products were further sequenced using the PCR primers to confirm the correct integration. Sequencing data using the forward primer shows the correct integration of the 5' end of the left homologous arm in the donor vector. The reverse sequencing primer confirmed the correct integration of GFP puromycin cassette. The original ATG5 gene was replaced with GFP. Origene offers genome-wide human and mouse CRISPR knockout knock-in kits. With these complete, ready-to-use kits can knock out almost any gene from the genome and knock in the GFP and puromycin cassette simultaneously. You can study the consequences of the gene disruption and potentially do a promoter study at the same time. Origene also has other functional cassettes to provide different fluorescent proteins, luciferase, or different selection markers. If you wish to have the CRISPR knockout kit created a different donor vector, please visit our website to request a custom project. To learn more, please visit the Origene website and our YouTube channel.
The next one.
The ability to insert or knock in a gene at a specific locus is a tremendously useful method for dissecting disease mechanisms. However, in previous years, the insertion of a foreign gene has been far too tedious and expensive for many labs to perform. Thanks to the recent development of CRISPR-Cas9 technology, gene knock-in experiments have now become a very popular technique in modern molecular biology. In contrast to the random integration events by the lentiviral system, CRISPR utilizes a guide RNA that directs gene insertion with extraordinary specificity. In particular, the AAV-S1 safe harbor site is often a preferred target site for gene knock-ins. This is because it enables consistent and robust transgene expression with minimal toxic effects. At this safe harbor site, the surrounding DNA maintains an open conformation, enabling stable expression of the newly inserted transgene. This makes safe harbor sites, such as the AAV-S1 locus on human chromosome 19 and the ROSA26 locus on mouse chromosome 6, very popular sites for targeted knock-in experiments. In this case study, we will walk you through how a red fluorescent protein gene was knocked into human embryonic kidney cells at the AAV-S1 safe harbor site. We will also demonstrate how the knock-in was verified using a PCR-based method. To begin, an sgRNA was designed to target the human AAV-S1 locus. Software analysis was performed to ensure maximum efficacy of on-target cleavage while avoiding any off-target effects. The resulting guide sequence was used to construct ABM's AAV-S1 targeting all-in-one vector. This vector expresses the Cas9 endonuclease under a CMV promoter and the sgRNA under a U6 promoter. To achieve the knock-in, a donor plasmid containing the gene of interest is required to provide a template for homology-directed repair. The HDR pathway is a mechanism in cells that repairs double-stranded breaks by using a DNA repair template homologous to the sites flanking upstream and downstream of the break site. To enable HDR-mediated gene insertion, the gene of interest must be flanked by such homology arms. Therefore, a second plasmid was constructed by cloning an RFP puromycin expression cassette into ABM's repair template plasmid for the AAV-S1 locus. The resulting AAV-S1 donor RFP plasmid had the RFP gene under a CMV promoter and a puromycin's resistance marker under a PGK promoter. Once both plasmids were ready, they were introduced into HEC-293 cells using ABM's transfection reagent, DNA fectin. Following transfection, the cells were passaged 10 times to dilute out the episomal donor vector. Following, the resulting HEC-293 cells were given 3 to 4 weeks to grow in the presence of puromycin to select for the cells with successful knock-in of the RFP puromycin resistance cassette. Cells transfected with both AVS1 targeting all-in-one vector and the AVS1 donor RFP plasmid were healthy after 4 weeks, and greater than 95% of HEC-293 cells were successfully expressing RFP. Control cells that were not transfected with the vectors died after puromycin treatment. PCR genotyping was carried out to confirm that RFP was successfully knocked in at the AVS1 site. In this method, 2 primers were designed. Primer 1, targeting the 5' homology arm upstream of RFP, and Primer 2, targeting a site within the RFP puromycin resistance cassette. Gene knock-in was confirmed when a PCR product of 1.1 kb was observed, indicating that primers 1 and 2 were both able to bind to the edited genome to enable successful PCR amplification. In the case of a non-successful knock-in or in the presence of wild-type control cells, no PCR products were observed, as primer 2 could not anneal to the target site and enable amplification. We hope this case study has demonstrated how the CRISPR-based gene knock-in technique can make your exploration of gene function amazingly easy and efficient. CRISPR-Cas9 mediated gene knock-in allows for biallelic modifications of some genes to be generated with ease, with phenotypic changes in mice observable in a single generation, an unprecedented time frame. Let ABM's team of CRISPR scientists help you knock in any gene into any cell line you want, all with minimal toxic effects at the AVS1 or ROSA26 safe harbor sites. Explore ABM's extensive portfolio of customizable CRISPR services and products today, and start knocking in genes, tags, markers, and more. To make it even easier for you, simply describe your desired knock-in, and we'll take care of the rest.
So we will stop it here today. I think that was the last slide. So I will share this slide with you. If it is not big. Oops. It is big. Hundred MB is the limit, and this is 110 MB. Give me just a second. Yeah. Some of the PCR methods, plant DNA extraction, all protocols I'm sharing here. I think I also have shared the book with you already or not. I think books have been shared. Otherwise, I will share it now. This is the best book for your molecular biology techniques. Okay, so that's it for today. We are done. Thank you very much. So you will get a lot of things every time when we are done with the lectures. So sayonara. On that note, I will leave here. So tomorrow is holiday. Weekend is a holiday. We see us on Monday now. So we're probably done with the very basics 3 days workshop so far. If you see, and we did a lot of topics. So go and read them and revise them during the weekend. Thank you very much.